Hey, yo, is this thing on? All right. So this is lac operon here, AP Bio level. We have lactose, a sugar in milk that has two hexagons. The one hexagon is galactose, the other one is glucose, and they are linked together with an ester bond. That's an oxygen bond right there. Okay, that's how lactose comes. That's in milk, that's in mammals. All right. So we can digest this with lactase. If you are a eukaryote and you are a human mammal and you are in your infancy, baby stage, before you have teeth, before you're eating, you can just make lactase and digest that in your stomach. Pretty easy. That's not what we're talking about here, interestingly enough. This is how a bacteria, an E. coli, is going to break this thing down. So if there's E. coli in your gut, they can break lactose down in this way. And if there is E. coli bacteria or other types of bacteria present in milk, presumably they could break this thing down. I don't know if this is how you make cheese or yogurt. I do know that's a lactobacillus bacteria. They probably have a similar lac operon because this operon works to digest lactose. So that's probably how it is. All right. So what we have here is DNA. Our DNA is a gene. It's got multiple genes in it. This DNA double helix here. And so they're showing you kind of the double helix. The rest of it is here. It's, thousands of base pairs long. So they're just kind of like showing us the basic part of it. Okay. So we'll start with this case where lactose is present. We've drank milk, and so now we have to digest the milk. Well, specifically the lactose in the milk, because there's multiple things in the milk, but the lactose is what we want to digest. So we're going to put RNA polymerase onto the promoter region, which is typically, a promoter is like a TATA box, T-A-T-A, which we kind of see right there, and it's trying to bind, but it can't bind if there's no lactose present. But in this case, lactose is present. We've just had milk. It's been converted to allolactose by this beta-galactosidase enzyme, which comes from the LAC-Z region of the lac operon. Now, so this is a gene. Genes are going to be expressed and transcribed, translated into proteins. That's just what gene expression means, okay? So to express this lac operon, we have to have milk present, which is really nice because if lactose is not present, then the repressor is active, this protein repressor is active and blocking RNA polymerase from transcribing the gene, which is great because we don't want to waste our time making these enzymes to break down lactose if it's not there. So for most of our adult life or any time when we are not drinking milk, even in times when the baby is sleeping, the baby mammal is sleeping, we don't want to digest this stuff. Oh, wait, hold on. This is bacteria, not humans. So just a quick reminder to me and you that this is how bacteria are going to do it. E. coli specifically is where we studied this lac operon. And so, but when the bacteria especially cannot afford to waste any energy, cannot afford to waste ATP here. So we need to make sure that they are chilling and not creating these enzymes when lactose is not present. But if you pour some milk on some bacteria, either they're in your tummy or maybe they've just landed on some milk, in some monk's cave to make some cheese or something, they're going to digest the lactose. Now, why is another zoom out thing. Why would they digest lactose? Because once you digest that lactose, you break apart the galactose and the glucose. You convert galactose into glucose because, as you see, they're outrageously similar, okay? And once you have glucose, then you can go through glycolysis. And if you are a little bacteria, you can make ATP and NADH out of that glycolysis process. But it's glycolysis, not galactosolysis or lactosolysis. So you have to convert things into glucose first, and then it can go there, all right? So now the nitty gritty. When lactose becomes present, it is converted into allolactose inside of the cell, okay? So lactose is able to come into the cell, likely from permease, because permease is an enzyme that allows things to permeate, and permeate means to enter, okay? So we have permease here, letting lactose in, beta-galactosidase, I looked up is what's going to convert lactose into allolactose. And we only need that conversion because allolactose binds to the repressor protein. Lactose will not bind there, okay? So if allolactose is bound to the repressor protein, you see that it changes the protein shape. That is kind of the deal with all proteins. If they are the right shape, they will function and do their job. If they are not the right shape, they cannot function and do their job, okay? Now, this particular protein doesn't really seem like an enzyme that's powered by ATP. It looks like it's just a protein. If it's the right shape, it will bind to the operator region, and that will block RNA polymerase transcription. However, as you see, once lactose converted to allolactose, binding, changing shape, cannot bind to operator. RNA polymerase has been floating around, waiting inside of the E. coli bacteria to land on this. Now, the reminder that DNA is just out in the cytoplasm cytosol of a E. coli bacteria because there's no nucleus in all bacteria, prokaryotes, no nucleus. So all this stuff is just happening within the cell. All right, so once we get RNA polymerase to bind, then transcription occurs. We're going to transcribe the LAC-Z gene. There's going to be a start and a stop. The ribosome will just ride along. It will latch on. It will start with AUG here, amino acid, amino acid, amino acid, amino acid, amino acid, amino acid, amino acid. Then it will stop. Then the ribosome will stay on because it will start again, AUG, and then a bunch of different amino acids, all those codons, 3 letters in the mRNA. Then we'll get to another stop. Then it will start again. Now that's the LAC-Z gene, which makes the beta-galactosidase enzyme. Then your LAC-Y, which makes permease. Then your LAC-A, which makes transacetylase, okay? Which is probably what's going to convert galactose into glucose. But, you know, that's going to allow us to digest the stuff, and then we can go through glycolysis. So your RNA polymerase is just going to keep running, because there's multiple RNA polymerases, okay? And so they're all going to land on here and just go boom, 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 boom. And they're going to make lots of different mRNAs as long as the repressor is not there, okay? Now, if the repressor is not on the operator, that's because there is lactose present, okay? So we're still trying to digest the lactose. The moment that we digest all of the lactose, then there is no more green allolactose because there is no lactose, and the repressor becomes active again, the correct shape, and it will latch onto the operator, and that is going to turn RNA polymerase off as soon as all the lactose is digested. So it's a perfect on-off system. It is inducible. We call this an inducible operon because it is normally off, but you can turn it on. The operon is off without the lactose. That's normal, okay? Suppose, right? And then when you need it, it turns on. This is an example of homeostasis, and it works just like your thermostat in your house, right? Like if it's the right temperature, your heat is not going to turn on. If it's the right temperature, your air conditioning is not going to turn on. But as soon as it gets too hot, air conditioner turns on. As soon as it gets too low, okay? But it is normally off. And then if it comes outside of the range, then it's going to come back down. That's called homeostasis, okay? So we can see a picture of this happening here where the lactose is present. It's been converted to allolactose. So this repressor right here cannot bond to the operator region. RNA polymerase binds to the promoter, TATA box, thymine, adenine, TATA box, and it will just keep going. Like I said, we're just going to make more RNA. Once the RNA has been created, again, this is just out in the cytoplasm of a bacteria, no nucleus, and so there's going to be a ribosome latching on, going ching, 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 over and over and over, because there's lots of different ribosomes, and there's lots of different RNA polymerases. So it's just going to keep going and going and going until all of the lactose has been digested, then there's no allolactose, then it latches back on, and we're all good, okay? So I'd like to draw this real quick so that we can be comfortable with this, all right? So we've got a picture of lactose there as what we're going to be digesting. The way that you would draw this is you have your promoter region, your operator region, you have LAC-Z, Y, and A. Now this is all part of DNA, so to remind yourself, you can draw that DNA double helix. Then you can label this the promoter, the operator, LAC-Z, LAC-Y. Y and lac A. How do you remember ZYA? I don't know. Remember, okay? ZIA, something like that. We got POSI A, sure, promoter operator ZYA. This is not as important for you to memorize, but it can be really helpful. Now, I almost forgot the other part, which is the gene that is going to make the repressor, which is over here, and they call that the LAC-I. LAC-I for inhibition, which is going to stop. And so this LAC-I region over here, LAC-I, is what's going to be transcribed, translated into the protein repressor. So we can draw a repressor just like that Lego repressor, and it will latch on to the operator. We can literally draw it latched on to the operator. Here's the repressor. Okay? Now, so in this case, you could label this no lactose in E. coli. Okay? Which means no transcription, translation, no enzymes, no digestive enzymes. Okay? So that's going to be our first case. Now the second case could be, maybe you could just draw it in a different color or something. This is when lactose is present. Okay, so lactose present, lactose is going to be converted into allolactose, and then allolactose, which we can label ALAC, is going to bind to repressor. If allolactose is present, allolactose is going to pull the repressor off. Okay? The way that we typically draw this is that if you have your repressor, which we could have done in a different color, your repressor in here, repressor, allolactose is going to inhibit the repressor. So we can draw a line and an X there, and allolactose is bound to the repressor, and you could even draw that the repressor is like non-functional, like a different shape. Okay, something like that. And you could say non-functional. Okay, now what this means is that then we get our other protein, RNA polymerase, to land on here. So we'll draw RNA polymerase, RNA poly, and we'll go this direction. And we are going to then, with our pink, we'll show that we will make enzymes, digestive enzymes. An arrow means that you're making something. A line with an X means that you're stopping something, inhibiting something. So if you're making digestive enzymes, they are going to break down the lactose. Okay, that's where we draw the line with the X, break down lactose. Now, I've not invented these like lines, arrows, X's. This is like how everybody draws it, okay? This is how you draw diagrams in biology, okay? So here you could write this transcription, translation, which is going to give us mRNA, which is what we saw in those pictures, and then eventually the ribosome, mRNA plus ribosome gives us our digestive enzymes. RNA polymerase is what gives us, so it was green, we can draw it like that, that gives us that when that's present, okay? Obviously this is kind of like a messy diagram, but I just want to show you the basics. If those diagrams don't seem easy, being able to draw this is a way to learn. You could also like write things out, but that's the way to do it, okay? Now, the other operon that is important to know is the trip operon, which is unbelievably similar. That once, I'm not even going to spend much time on it, because once you know lac operon, you can just say trip operon is the opposite. So with the lac operon, we don't want to be digesting lactose if it's not there. But with the trip operon, we do want to be making tryptophan. Tryptophan is an amino acid that is beneficial or necessary. Tryptophan right there, okay? Tryptophan, UGG codon, gives us tryptophan, okay? And that is necessary for making a whole bunch of proteins, right? And you need to be able to make that because it is not usually in your diet, but okay, so you have to have this. So if tryptophan is absent, like we don't have it, then we need to be making it, operon on. So the repressor is inactive. So you see our shape for our repressor, our protein. TRPR makes the regulatory, that's the regulatory gene, which makes the regulatory mRNA, which makes the regulatory protein, which is your repressor. And it's not active right now. So we know the repressor is inactive. The TRP operon is on, okay? See how this is opposite from lactose? We want to always be making this stuff if we don't have it in our system, because we always need to be making proteins. And again, this is likely from an E. coli bacteria. So the bacteria needs to constantly be able to make tryptophan as an ingredient to making other proteins, okay? So RNA polymerase binds to promoter, right through the operator, and kaboom, 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 kaboom. There's just 5 there instead of 3 with the lac operon. Then you're making a bunch of subunits, okay? So first you're gonna put the amino acids together. That's gonna give you your primary protein conformation. Then they're gonna twist up into alpha helices and beta pleated sheets with hydrogen weak bonds, and that's going to give you your secondary conformation. Then your tertiary conformation is when you zoom out and see the 3D, and that's going to be, this is at your tertiary conformation. Now, when you put A, B, C, D, and E together, they are going to form a full subunit, or a full, those are all subunits that will form the quaternary conformation, the full enzyme that can make tryptophan. Because tryptophan is a very small thing, right? But the enzyme factory that's going to make it, that's what we are making here, okay? So a couple of different steps with that, okay? But it's a similar kind of a thing. Start, stop, and then it's just going to go and go and go and go all the time, okay? Like for the whole bacteria's time. Now, it will only ever turn off if tryptophan is present. So if the bacteria comes upon eating some tryptophan, that's a tiny little amino acid, and the bacteria is like, yay, let's turn off the tryptophan making enzyme, like factory, okay? Because we just ate some, we got it, okay? So tryptophan is literally going to turn its own synthesis off, which is perfect, right? So tryptophan will bind to the protein, repressor will change into the active form, and then it will bind to the operator region, blocking the RNA polymerase, and no RNA made, which means no enzyme subunits made in the tertiary conformation, no giant megazord quaternary conformation enzyme to make tryptophan, right? And none of that, none of that, none of that, and because clearly the E. coli has enough. Now, as soon as the E. coli uses that tryptophan to make proteins over, I don't know, the next couple of hours, days, whatever, then at some point the E. coli will run out of tryptophan, and then it will want to be turned back on, okay? Now, this might be the case in human beings, eukaryotes, we don't know, okay? But then it will be able to turn it back on, all right? Now, before you go thinking, oh my God, is he gonna throw another example at us? No, these are your 2 examples. Okay, this is what AP Bio wants you to know. This is like your first couple years of, biology, right? Like, as long as you know these are the 2 concepts. The lac operon is inducible, which means that it can be turned on, whereas the tryptophan operon, the TRP operon, TRP, is going to be called repressible, okay? Because it's always on, it could be turned off. Repressible means it's always on, but it could be turned off if you've got tryptophan present. The key difference is that lactose is a sugar, which is going to be utilized for making energy, glycolysis, and tryptophan is an amino acid, which is necessary for synthesizing proteins. So if you can understand those bigger picture zoom out concepts, then it will make a lot more sense to say like, all right, so if we've got this energy source, let's consume it, let's turn on the stuff and make it, okay? If we have the energy source present, then let's turn it on so we can digest it and we can get energy from it. Whereas with an amino acid, it's like, okay, we always need to be building that building block, okay? We're always making proteins. But if we find the building block somewhere lying around, literally in the food that we consume or the food that the E. coli consumes, then we don't need to be making it at that time. Okay, you find some free bricks, you can make a wall. Okay, until you run out of bricks, and then you got to start making more bricks. Okay, all right.